Hello friends, vanakkam. Welcome to Whiteboard Learning with me. Let's discuss about red blood corpuscles or RBCs. Red blood corpuscles. This is the structure of red blood corpuscles. So this is biconcave disc with no nucleus. Carry oxygen. So RBC carries oxygen, but it is having no nucleus or without nucleus. This is shaped biconcave disc shape. So this is the cytoplasm containing hemoglobin. Next, how to identify the slide. So we identified the slide and write the identification like the given slide is identified as RBC or red blood corpuscles. So this is the diagrammatic view of RBC. Side view. This is the side view and this is the top view. In side view, it's having 2 micrometer. Then in top view, it is 7.5 micrometer in size. Then red blood corpuscles or erythrocytes are responsible of carrying oxygen from the lungs to tissues and maintain pH level of blood. So this RBC or erythrocytes, it is responsible of carrying oxygen. Already we know it is carrying oxygen from the lungs to tissues of the body, all body parts, every tissues and cells, and maintain pH level of blood. Then so the explanation of slide red blood corpuscles or erythrocytes. First point, the red color of RBC is due to the presence of a respiratory pigment, hemoglobin. So hemoglobin is the respiratory pigment. It is red in color. Then hemoglobin plays an important role in the transport of respiratory gases. It is important role to the transport of respiratory gases. Okay, important role in the transport of respiratory gases. This is second point. Third point, RBCs are produced in the red bone marrow of large bones and are destroyed in the spleen and liver. Then final point, the average lifespan of RBC in a healthy individual is about 120 days. Then hemoglobin. This is the heme particle. This is the hemoglobin molecule. So this is the oxygen molecule or molecules of oxygen. This is the hemoglobin molecule. So this hemoglobin bind with oxygen molecule, that is called RBC, red blood corpuscles. Then oxygen binding hemoglobin is named by oxyhemoglobin. Then this hemoglobin without oxygen, so oxygen being released from hemoglobin. Then when a hemoglobin combines with oxygen is called oxyhemoglobin. Then each cytoplasm of an RBC contains 270 million molecules of hemoglobin. Then hemoglobin is a protein that contains iron and gives the red pigment to our blood. Then this is the test tube. So we take the sample of blood in test tube and shake well, leave it a few minutes. After we see these three layers. So this sedimentation is erythrocytes. This middle layer is leukocytes or platelets. Then this is the plasma. So red blood corpuscles or RBCs from a major component of blood. So see, heme is the part of hemoglobin. Hemoglobin is a part of red blood corpuscles. Red blood corpuscles contain hemoglobin, which actually gives the bright red color to these cells because of the heme group. Next, gene degenerative forms of RBCs. So this is poikilocytosis, anisocytosis. So this is the abnormal forms of RBCs. So variation in shape depending upon shape abnormality. It's divided into these categories. This shape is acanthocytes. This is target cell, ovalocyte. This is stomatocyte. This is sickle cell. So this is the RBC sickle cell formation. It leads to anemia, sickle cell anemia. So this is the abnormal variation in shape, that is called poikilocytosis. Then depending upon size, it varies. This first, cystocytes. Size is being 2 to 3 mu in size. This is microcyte. This size may be 5 to 6 mu. Then normocytes 7 to 8 mu. Macrocyte 8 to 12 mu. Megalocyte 12 mu. Then this is also the abnormal forms of RBCs. Abnormality of RBCs. This is echinocytes, lymphocytes, poikilocytes, cystocytes, sickle cell, teardrop. Abnormality of color. This is the form of RBC abnormality. This is color of RBCs. Dimorphic, hypochromia, polychromasia, spherocytes. Then abnormality of membrane, agglutination, HPC pathology, stomatocytes, target cells. Then abnormality depending upon size, anisocytes, macrocytes, microcytes. Inclusions: Howell Jolly bodies, parasitic infections, punctuate basophilia. This is the other abnormal forms of RBCs. Then this is polycythemia. So polycythemia means abnormal rise in RBC. It is called polycythemia. So abnormal rise. This is normal blood cell. Then this is abnormal rise called polycythemia. Then anemia, this is anemia, means when the drop in RBCs, it is called anemia. So the RBC level is very slow, or it is abnormal drops in RBCs. Then a decrease in the RBC causes a decrease in oxygen, which pushes the kidneys to secrete erythropoietin into the blood. Flowing of RBCs, this is less deformability of RBC, but the high deformability RBC is a healthy RBC is going through this spot. Serious problem in microcirculation. Due to the microcirculation process, less deformability RBCs. This is hardened RBCs. It is not through going to microcirculation, but the healthy RBCs going through and involve the microcirculation. So this is the micro view of RBC. Biconcave disc. This is the sectional view. So see, potassium, sodium, chlorine ions, ion channels. See, ankyrin, actin, band 3, spectrin. So this is elongation, this is tank trending, swinging, tumbling. So cytoskeleton, intracellular viscosity. So this is the microscopic sectional view of RBC. Then the hemocytometer. This is the hemocytometer. It is used to calculate the RBCs and blood cells, white blood cells, red blood cells, platelets. All blood cells are counted by hemocytometer. The number of erythrocytes in the blood is counted using hemocytometer. So this is the hemocytometer. So Newbore's chamber or hemocytometer. This is the important chamber of hemocytometer. So this is the part of the slide. Glass cover, mounting support, 0.1 millimeter gap, hemocytometer slide. This is hemocytometer slide. This two part is main part to count the RBCs. So 9 large squares. Each large square is divided into 25 medium squares and 16 small squares. This is 1 millimeter, 0.25 millimeter. These boxes are counted by red blood cells. So this is the red blood cell counting portion in hemocytometer slide. Totally this slide is being 3 millimeter in size, 0.5 millimeter. This each one is divided into, see, 4 columns, upper 4 columns, lower 4 columns, top and sectional view. This is 0.05 millimeter in size. This is the RBC countings. Then hemocytometer is an instrument that helps in measuring the level of hemoglobin in our blood. Okay, this is the hemocytometer slide. Say this is 1 millimeter, this is 1 millimeter. This portion, this portion, this portion, this portion are very important portions. This is also 1, 2, 3, 4, 5. So A, B, C, D, E. These are the portions to counting blood cells. So this is the hemocytometer. Another points. The large center square is used for RBC and PLT counts. The center square is divided into two smaller squares which are each subdivided into 16 squares. Only 5 of the 25 squares are used to count red blood cells. These 5 are usually the 4 outer squares and the innermost center square. The entire large center square is used to count platelets. So this red points 1, 2, 3, 4, 5. These 5 points is very important points. These areas of the grid where RBC are counted. Then a drop in RBCs can occur because of blood loss, less blood formation, or destruction of RBCs. When seen individually, red blood corpuscles are yellowish due to a pigment called bilirubin. Okay, this is one of the disinfection leads to jaundice. So this is something about red blood corpuscles. Thank you for watching. Please subscribe my channel, share my channel. Thank you.